From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on March 20th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 84th episode of Matters Microbial. Thanks as always for listening, viewing, and sharing the good microbial word. It really matters to me. I had a couple of emails and comments about my examination of water samples recently. First, I'll keep my discussions of my own work short. I know you want to hear from our guests. I'm thinking of starting a Substack site to share this kind of information if people are interested. Second, please remember that we live in a microbial world and we swim within a veritable sea of microbes at all times. Here is a bit of artwork created by a former micronaut in my microbiology course many years ago, the Peanuts character Pigpen surrounded by microbes. Because you see, we are part of a diverse and crowded microbial universe, and honestly, only a tiny number of these organisms can cause disease, despite how the press shows things. I'm reminded of this daily as I look at a nearby bookshelf in my office, modifying some of the plush toys from Giant Microbes, the company. When I look at my students' water bottle buddies from their reusable water bottles in my classes, I see organisms such as you can observe in this word cloud. To be sure, there are some organisms with genus names that might concern you, but mostly they're related to breaking down plastic, living on small amounts of nutrients, and overall forming biofilms so they're not dislodged over time. Please see the article in the show notes on this topic. It was suggested by my producer, David Renata. Let's have a quick look at another site I am microbially assaying, a drinking fountain. As you can see, here is the site that I swabbed from. Now, many microbes will be trying to make a living there based on dust, low nutrients, the need for biofilm formation, etc. We had very little to do with it. The initial swab plate on R2A showed a limited population in this water fountain, as you can see. When we go up to a close-up of that plate, we see three different colonial types of microbes on the R2A plate: white, orange, and red. Now, I'm pretty sure I know who each of them are, but time will tell. I did my best to isolate each of them on R2A plates for further analysis. Isolate number 1 gives rise to white colonies, as you can see. Isolate number 2 gives rise to orange colonies, as you can see. And isolate number 3 gives rise to these tiny red colonies, as you can see. They are beautiful. I cannot wait to share more data with you all now that they are successfully isolated. And please remember that only about 5% of the microbes present in this kind of environment can even grow on nutrient media in the lab. It is indeed a microbial world, listeners and viewers. Never forget that. In recent years, we've all seen how wastewater could be used as a measure of viral prevalence, not just SARS-CoV-2, but also influenza and other viral diseases. In fact, wastewater can tell us a great deal about the spread and prevalence of many kinds of microbes, ones that cause disease as well as those that don't. In addition, it is even possible to examine the DNA within wastewater to determine if particular genes are present, for example, genes for antimicrobial resistance that might be spreading to other bacteria. So, I'm delighted to have Dr. Rachel Poretsky of the University of Illinois Chicago on the podcast to discuss this and related topics. Welcome to the Quality Quorum today, Rachel. Wonderful to see you again.
Thanks for having me. I'm super excited to be here.
Oh, and you just got done teaching a class. Are you still vibrating from it?
A little bit. It's always an exciting moment to come out of class and be able to share all this information with a next generation of learners, as you
Oh, it's
know.
and when they actually come up and ask a question that isn't, is this going to be on an exam? I live for those moments, don't you?
100%. Yeah.
What was your topic today, by the way?
We talked about anaerobic respiration. So we talked about metal reduction and denitrification.
Breathing metals. It's a big deal.
Amazing. Yeah.
Yeah, I'm really happy about it. And thank you so much for being able to rush upstairs to your office to join us on the podcast today.
My pleasure.
So, by the way, I love your microscope behind you. I'm going to have to zoom in when I look at pictures later of the other artwork. As has been said many times, I have too much in my own background, but
Yeah.
it's okay.
I have
Any,
academic artwork and children artwork.
yeah, I have some. I have Michelle Banks right here.
I do too. Michelle Banks. I
There
can't
you go.
there.
3 cheers from Michelle Banks. Buy her artwork.
Yes.
So I think a good way to start is to tell listeners and viewers what wastewater is, because we all think we know what it is, but I bet we don't.
Yeah, wastewater, well, it's what it sounds like, right? So any waste that you put down the sink, the shower, the toilet, not just feces and urine, but really your soap, anything that you wash down the drain will end up in any major city, will end up in a wastewater treatment plant. And how the wastewater treatment plants process those samples or that material really depends on how many people it's serving and what type of mechanism it's using to treat those samples. But the principle is to remove anything that could be cause harm to the public, because once it gets treated, whatever it remains is called effluent, and that gets discharged into the environment, usually like a lake or a river or a nearby body of water. So that it has to be cleaned and processed in a way that removes chemicals and removes whatever is regulated in wastewater.
You know, it's interesting, Rachel, because when I first started thinking about this a million years ago, I didn't understand that dissolved nutrients and dissolved nitrogen compounds are also pollutants because they lead to things growing uncontrollably near the effluent. Is this true?
That's right, and in fact, there are a number of things that are regulated about what wastewater treatment plants can discharge, and this is regulated at the federal level. And among that is nutrients, also disease causing or enteric bacteria, but there's a lot that is not regulated, and so plants tend to focus their designs to remove certain specific microorganisms and nutrients. And then whatever remains is what gets released and is deemed safe because they've managed to remove all those, that list of chemicals and organisms that they need to process.
You know, it's so funny because the thing that I would think about toxic chemicals as being a problem, and I would think about living organisms. Right. And in fact, and this is not a topic for today, but
Yeah.
wastewater treatment plants are usually bioreactors, and they use microbial life to remove that additional nitrogen dissolved in the water, that additional carbon that's in there.
Right.
And
So it's a really interesting, sorry to interrupt, but that's a really interesting um, point, is that it is a microbial process. Whether some plants use an activated sludge treatment, some use a biofilm, a trickling biofilm system, but there's all sorts of important. We talked about in my class Animox recently, and so that's an important process that's used in wastewater treatment, a microbial process. And so the whole treatment process is a mixture of settling and filtration methods, so abiotic, but also the biological treatment.
I will never look at tap water the same way again, is the take home.
Well, hopefully, in most places, it's completely separate systems, not connected to drinking water. But That brings up an important point, which is here one of the reasons we started looking at wastewater even before COVID was because Chicago, where I am, we get our drinking water from Lake Michigan, and the story in Chicago is that the Chicago River used to naturally flow into Lake Michigan, and that's where before there was sophisticated, 100 plus years ago, before there was sophisticated wastewater treatment, wastewater would be discharged into Lake Michigan, which is where the city got their drinking water, and people were dropping dead of cholera. And it took some thinking to be like, maybe we shouldn't be drinking the water where we put our waste. And so the Army Corps of Engineers came in and reversed the flow of the Chicago River to flow away from Lake Michigan. But today we still have many wastewater treatment plants that discharge treated but still wastewater, so treated effluent, into Lake Michigan. So in all cities that surround Chicago doesn't do this anymore, but lots of other cities still discharge into Lake Michigan, and it's where we get our drinking water. So I think it's really, there are places where wastewater and drinking water are connected, even distantly.
It's interesting to me when you talk about the processing of these things, because I was always told that one of the best places to look for diverse bacteriophage is in wastewater effluent.
Mm.
And presumably that's due to the huge bioload of the bacteria and archaeans that are actually helping with this process. At least I tell myself that. But now we're thinking, maybe that is a situation with high loads. Then you talk about what's in pipes and such, and you find out some of those situations have to do with low nutrients as well.
Mm-hmm.
And all of these are different environments where microbes are trying to make a living, is the take-home lesson.
Exactly. And I think there's so many interesting dynamics. I work with modelers who help us try to understand how organisms go from like biofilm to suspended growth in. You had a guest talking about the built environment recently, Erica Hartman, right? And so I think, in pipes, there's different levels of microorganisms and how they decide, right? How they decide to be planktonic or in a biofilm is really important for any downstream effects.
This is one of the things that I battle with a lot, is the negative viewpoint that many people have about the microbial world. And so when we talk about food safety, water quality, things of that nature, I could understand the concerns that people might have. But at the same time, the things that interact with us are at a relatively low level overall, and they can be really terrible, but it's just a tiny number. In fact, Maggie Middleton, known as Vexed Muddler, once put together a cartoon about pathogens as kind of like gang members from the 1950s riding motorcycles in leather jackets. And I always like that because they get all the press when in fact most microbes could care less about us.
Yeah, and I think that's where our work with wastewater has sort of ended up. We're looking at a balance of the pathogens, which I think tend to be, like you said, in the minority. Like most microbes are not going to cause disease in humans, especially. But when they do, it's a big problem. But on the flip side, we can use wastewater to tell us about some of the pathogens that may be circulating in the environment. And so there's and then all the dynamics in between, the interaction between the organisms that we contribute to wastewater, the organisms that are in the treatment process, how they end up in the water and how they interact with each other there, and how they inform us about just health of the ecosystem and health of the human community surrounding.
And you were a microbial ecologist.
That's right.
That's right. And so what a lot of people don't appreciate is how difficult ecology really is, because it's the interactions of many things together. And we're reductionist by nature. We like to look at things by themselves, and that's not how anything lives.
Right.
Not even us.
Yeah, I often have people ask me as a microbiologist, what organism do I study? And I'm like, all of them, whatever's there. And it's really more about how they interact with each other and interact with their environment than one particular organism and what it does, which is also valuable to understand, but not what I do.
No, it's wonderful to hear this. So let's go on. So this is what made you think about working with wastewater, is the idea of how different things end up in different places. I'm sure you did transects initially, trying to find out what was being released and where it showed up. What
Mhm.
were some of your first efforts?
So I I got interested in I had done my PhD in marine science, so I had studied um, microbes in the ocean previously, and then I did a postdoc at Georgia Tech where I was in an environmental engineering department, and we were looking at degradation of contaminants by microorganisms, and it was really from like more of a little bit of an engineering perspective, and part of those that contaminant question is from organisms that are introduced into the environment through things like wastewater. And so when I started my position here, it was actually a really good timing for me to start looking at wastewater in Chicago because it was right after the EPA had designated the Chicago River as a recreational water body that needed to conform to recreational water quality standards. And before that, we were the largest city in the country, maybe even in the world, that didn't disinfect our wastewater before releasing it into the environment. And so this switch was supposed to happen like a year or so after I started my position here. So I was like, great, let's see what organisms are here. Let's see what organisms are being released into the environment before disinfection. And let's see how disinfection changes that, because disinfection is just the last step after all the processes in the treatment plant that help remove, like we said, nutrients and organisms. It's a final treatment, which is either like chlorination or UV treatment or something else. And so that's where I started doing wastewater and trying to understand what was there and how it changed when uh, there were big rainfall events that introduced runoff, or what we have in Chicago and many other cities in the U.S. Is these combined sewer overflows, where when it rains a lot, you get untreated sewage entering the environment. And so, yeah, we found evidence of antimicrobial resistance genes being introduced into the river. We found that disinfection doesn't kill everything, which probably shouldn't be that surprising when we think about it, that there's lots of organisms that are resistant to all sorts of exposures. But we found genes and organisms. In fact, we found RNA, so like active organisms that were still present after disinfection. And I think, the purpose of the disinfection is to remove pathogens, and I think it does a good job at that. And treatment certainly reduces the number of organisms and the number of genes, but there's still stuff that gets through no matter what happens.
So really quickly for listeners and viewers, there's a certain degree that is really important to remember about what was just said. The first thing is antimicrobial resistance, which we've talked about on several episodes of this podcast. The idea that some bacteria can become resistant to the antibiotics that we've come to rely on. And this is a double-edged sword, double-edged pilus. I don't know quite what to call it, but you get the idea. So the trouble is that bacteria and archaea really are, evolution works on them all the time. And it's a wonderful system for studying evolution for this reason. So you'll get bacteria that are resistant to antibiotics, and there are 4 different ways this can happen, not relevant to today. But sometimes those genes can be transferred to other organisms that were originally sensitive to them. And
Mhm.
that's what is so interesting about the work here, because this idea behind horizontal gene transfer, one form of it is simply called transformation. And if you're seeing the genes, that means that there's DNA out there that could potentially be sucked up like spaghetti by some microbes. There are other ways that horizontal gene transfer can occur as well. Additionally, I would be really interested to learn what would happen with low amounts of UV light and low amounts of chlorine, so that you actually will shape the community toward UV resistance and chlorine resistance. A lot of mycobacteria are highly resistant to chlorine in the water, for example.
Yep. Yep. And some of these treatments also change the chemical composition as well. So not just the microbes, but exposure to UV may alter chemical structures in a way that promotes evolution of other microbes too.
It's amazing.
Yeah.
I have a saying that the hand of Darwin is on all that lives, and it's really true.
Yeah.
We all study it in the lab, even when we think we don't.
Yeah, if I could show a figure, this plot of sewer bacteria that carry antimicrobial resistance genes really emphasizes the point that you just made. Which this is from a project that we have looking at sewers at hospitals, these long term acute care hospitals where you have typically have very sick patients who get exposed to all sorts of infections that can and these antimicrobial resistant infections can be really bad there. And so we actually did some all sorts of work with these wastewater samples. But one of the things we did was culture organisms for resistant bacteria. And we have a whole series of organisms that were able to grow with different resistance genes. Their resistance to specifically carbapenemases, which is a class of antimicrobial. And we find that there's organisms like the third, fourth one down, and somewhere on the y-axis, and then third or fourth from the bottom, Enterobacter cloacae is an example. These are organisms that cause infection in patients in the hospital. But then we have a whole bunch of organisms on this list where we find we can grow them out of wastewater, and they have antimicrobial resistance genes. And in fact, the same resistance genes are very close relatives of the resistance genes that we see in patients, but in what I think of as sewer bacteria.
Mhm.
So, we see infections by Klebsiella and Enterobacters, but we don't typically see infections by Acinetobacter or Aeromonas that often. There's some Aeromonas on here that are what I think of as typical sewer bacteria. And we think what's happening probably is that, maybe it originally comes from patients and comes from the hospital, but once it gets into the sewer, there's all sorts of resident microbes that live there, and that horizontal gene transfer comes into play. And some of these genes I think of as promiscuous, like more liable to be transferred horizontally and end up in whatever organism. And if we're trying to use this information to tell us what's happening in the hospital, it can be tricky because once it goes from the hospital to a complex microbial community, these genes can end up everywhere.
I have always been worried. I don't know how you feel about oysters, but I have always worried about filter feeders and like effluent and transferring antimicrobial resistance genes for many, many years. And I think some people have tried to look at that, but it would not all at all surprise me, because anytime you have large numbers of bacteria close together, they're going to be donating and receiving DNA. That's just the nature of it.
Yeah.
And I love the term promiscuous that I've found in recent years I have to be a little cautious of in the classroom, but it's absolutely true. I've tried to say, please forgive me this. The level of genetic promiscuity is like you having children with your Caesar salad.
That
And
makes
it is 100% true that these mobile genes, these easily transferred genes, they're not, there is an expression, dispensable core genome, that sometimes we
yeah.
teach about, but it's amazing the things they can transfer, and it's just always moving around.
Yeah, I hadn't planned to talk about it, but since you brought it up, I'll mention briefly that we started looking at mussels that live in the Chicago River, and we've been sampling for about a year now, almost exactly a year. We sampled over winter, and we collect mussels, both native and invasive mussels, and we're looking for organisms and antimicrobial resistance genes, and even organisms that are able to transform contaminants, because we know this is possible. There was a paper about a year ago where folks found antimicrobial resistance genes in mussels in Lake Michigan. It's you're absolutely right with these filter feeders. When you're concentrating water, you're concentrating whatever's in the water too.
I would cook your food. That's
Yeah,
basically
yeah.
that's basically my take home. Sorry, I don't mean to be a hater, and just that's what it is. I ate my share when I was younger too, and then Woods Hole cured me of that because
Okay.
I tried to study that many years ago.
Yeah.
In any event, that's something really important for everyone to take away is that level of horizontal gene transfer is everywhere and how it can cause problems. And a delicate point is, for the most part, this has nothing to do with just plain antibiotics. We just happen to see those events when antibiotics are applied.
Right.
Other things are being transferred too. It's like a network.
Right. Yep.
Fascinating stuff. You also mentioned that you're culturing things, and I don't know what proportion of, and by the way, we need to come up with an omics having to do with the sewer. Okay? I'll think about it. Sewer omics is too easy.
There was a paper, I don't think I could say it on the podcast, but it was in the title of the paper and they called it the SHITOME. Um,
Yeah.
that's not what I would advocate for, but yes.
Yeah. I have to share that with Jonathan Eisen,
Yes.
who has an antipathy toward omics terms. So I mean he'll like that one a lot.
Yes.
But it's interesting to think about what percentage of the sewer microbiome are cultivatable on nutrient media versus things that you can't grow in the lab. And most environments that I look at, this may be different with the sewer things. There appears to be a relationship between copiotrophy, liking lots of nutrients and being able to grow on certain lab media. Have you found that to be the case?
Yeah, we don't do a whole lot of culturing, and when we do, it's for very specific purposes. So these resistance genes and resistant organisms were cultured on a very specific media that
Right.
is selective for antimicrobial resistance. Um, I think, but I think you're right. Like that there's some that are going to grow on nutrient rich media and some less so, but that's part the reason that we do so much molecular work
Right.
and so much metagenomic and targeted sequencing, because that way we can avoid all of these pitfalls or challenges. Or as amazing as culture is and can tell us things, we I feel like we can get a whole lot more information when we just do large scale sequencing.
No, I can't agree more. And there's a certain aspect of the discussion of culturability that turns into what I call a quasi-religious argument. And I like what you do because you're looking at it from 30,000 feet and at the same time you're parachuting in to look at very specific. So the idea of having to cultivate something may be an issue in terms of ecology anyway, because some microbes can only grow in the presence of other different microbes.
Exactly.
And this makes your work even better for that. So who would know that there would be a whole series of organisms adapted to live under sewer conditions? But when you think about it, it has to be that way.
Exactly. Exactly. And so, a lot of our wastewater surveillance work depends on looking for just the genes. And then it gets tricky when they're genes that are more easily transferred because we don't know who the host was. Or if we're looking for viruses, it's near impossible to tell if that SARS-CoV-2 that we detected or that influenza that we detected came from an animal or from a non-human animal. Or yeah, so I think that there's balances to being able to take different approaches to try to understand a little more about the, like, zoom all the way out and get a sense of everything versus zoom all the way in and just know what gene. And sometimes it's sufficient to know what genes are present. And for most of our surveillance, our wastewater surveillance work, we're just looking for specific genes that are carried by pathogens that we have decided to monitor, or antimicrobial resistance genes, and not really caring, or I shouldn't say caring, but not as important to know which organism was the host.
I think that's really important because no matter what you're talking about, if you talk about a disease causing bacterium, for example, you can look for genes that are associated with that organism. I presume you could even look for their ribosomal RNA to have some idea of their prevalence. When it comes to AMR genes, they can be in a wide variety of organisms, and you hit the nail on the head bacterially speaking, it's really hard to determine in what organism that is necessarily.
Mhm.
Sometimes people can look out in terms of sequence and figure out, and that's what metagenomics can help you with, presumably.
Right.
Um, Although if you have lots of different copies of that AMR gene, that would be difficult, wouldn't it?
Yeah, and we've encountered that too, like trying to, because what we're doing is not just the presence absence, and I can actually walk you through our
Please.
how we do this wastewater surveillance.
Oh, so much. Yeah.
But what happens is, so we collect the wastewater either from sewers, manholes around, mostly around the city of Chicago, or wastewater treatment plants around the state. We currently sample in about 80 different cities around the state, which represents close to 70% of the state population that we're representing by wastewater that we're testing in my lab. And then we get those samples in the lab and we're quantifying. We're not just looking if these genes are there or not. So we first we have to take a a relatively large volume of wastewater and concentrate it down into very small volumes that we can work with on a large scale. And so small volumes on a large scale, we have a lot of automation. We put the samples into plates that get aliquoted by a robot, a liquid handler. But at the end, what we're doing is quantitative PCR, and we're using a type of quantitative PCR that's digital PCR. That's really cool. We have these plates that partition our samples, the nucleic acids, into about 12,000 different partitions. Each partition does its own PCR. And so at the end, we're counting how many copies of these genes are there. And the reason that I wanted to bring this up now is to your point that if there's multiple copies of a gene, that gets tricky. How those genes get, those pieces of DNA get partitioned, that can get tricky. So at the end, we're saying that there's this many copies of a gene that's in a nucleocapsid gene of SARS-CoV-2, or hemagglutinin gene of influenza. And so we're using those as a proxy for what's happening in the community, knowing that there's challenges with copy numbers and things like that. And that's why we use all this data modeling and interpretation of the data and matching it to clinical surveillance and other forms of data so that we can kind of make sense of it, because it can, again, when we're dealing with these complex communities, it's not always straightforward.
No, no. And, one of the things, and I want to make this clear, when we're talking about bacteria, we're talking about metagenomics. We've talked about all of these things a fair amount on the podcast. Viruses can be a little bit different, as you mentioned. And especially RNA viruses, you can have defective copies of things.
Mm-hmm.
And I don't know how you can sort that out very well, except if you get a hit, you get an amplicon, that there has to have been that gene present.
Right. And a lot of what we do is look at trends. So we assume that even if these events happen that make the data interpretation hard at a single point in time, if we're looking over a long period, then hopefully those same events are going to be fairly consistent over time. And so we can see that relative to December or relative to another point in time, it's higher or lower. And those are the types of data that we think about more.
To be very clear, I've never done any research in my life that benefits humanity.
Ah,
Okay. Just to be 100% clear. But in a lot of ways, and I'm not simply talking about the pandemic with COVID-19, where having some idea of what's going on from the wastewater, perhaps to detect whether or not there's going to be a surge taking place, that kind of thing.
yeah.
We all have seen the graphs at one time or another about that. And it must be very rewarding to know that the work that you're doing might be helpful to healthcare professionals.
Yeah, it's so strange that my career has come to this point because, like I said, I really came from a oceanography and understanding like carbon dynamics in aquatic systems to doing this very applied microbiology. But it turns out that there's so many things that are that translate and are consistent. And I've never done work in my career until I started doing this wastewater surveillance, where people are looking at the data that comes out of my lab the day after it's generated, and that there's multiple eyes on it, and it is satisfying. It's satisfying that it can be useful. It's satisfying. I have collaborations now with clinicians, infectious disease doctors. I'm now involved in this NIH sponsored bacterial and viral bioinformatics resource center, which is a tool that was developed specifically with an eye on pathogens and outbreaks, and making data analysis accessible. So we're making data accessible, and then we're
Mhm.
building ways to have people be able to look at data, the vast amounts of sequence data. As you know, like people are generating sequence data on a scale that's every year we say unprecedented relative to the year before, and so this makes it accessible for people to analyze those complex data sets too, and look for things like pathogens and antimicrobial resistance.
So I have to come up with a motto for you, and I think the unprecedented is to be expected.
Ah! I like it.
Please find a way to use that because it's true. You're finding out things. The patterns are what's important, and I've been waiting for a long time uh, to see ecologists, especially ones from outside epidemiology, start to look at these issues.
Mhm.
It's my belief that we actually learn more when people from outside areas come into it. It's how molecular biology was born, all these quantum physicists that wanted to get out of that field, right?
I think you're right. I think there's been in the last several years an increasing awareness of the fact that we need to understand whole community dynamics and not just a single pathogen or a single organism, because when you see one thing, there's all sorts of downstream effects, and there's all sorts of other things that need to be considered. Even when we use therapeutics, there's benefits and trade-offs that are involved, and if you are looking through a narrow lens at one specific organism or one specific um, situation, then you're not going to be able to predict what's gonna happen on a larger scale.
I can't agree more. And one of the things about this, and I want to be very clear to listeners and viewers, it's really common for people today to look at things that occurred in the past and see it in a very jaundiced way. I think we have to look at people like Pasteur and Koch that kind of started the idea of single isolates and organisms on their own. That was necessary to set the stage,
Mhm.
and now we can build on them. In fact, you'll laugh when I was when biofilms first became a thing, because that's how old I am. Um, there I would say things to people like, "Well, you're just looking at one particular type of bacterium forming a biofilm in plastic,"
Mhm.
and they say, "Okay, that's not fair. We're now using three." And I would just throw my hands up in the air because that's not really how microbes act in the real world. But if you want to study things, you have to start simple and build on those things.
Right. And
And,
and
and
on the flip side though, like I think it's also important to characterize organisms really well, which we can do now with both advanced sequencing tools and analytical tools, is look at the genome level. I go like this when I say
Mhm.
genome, right? But you can look at the genome level, and we can capture things like evolution, viral or bacterial evolution, almost in real time. And so it's not. I don't think it's mutually exclusive. Like we have to. I think we have to do both. People have asked me if what we're seeing with what with the new variants all that keep emerging of SARS-CoV-2. I mean, we sequence hundreds of samples a week, um, from wastewater and do whole genome sequencing of the virus that causes COVID. And we have seen all the variants and all the sublineages that exist, and then some. And people ask me, is this evolution unique to SARS-CoV-2, or do other viruses do this? And I say, sure, other viruses do this, but you know what? We have never sequenced something this deeply for this long to know this. And so I'm sure lots of other bacteria and viruses show the same sorts of evolution and divergence over time, but we're I feel like we're still in the infancy of genomic surveillance of pathogens, and kind of, I don't want to say we lucked out with COVID, but we took this opportunity to really grow this discipline of being able to look at organisms really deeply, and be able to see what drives the evolution and how does that impact its transmissibility or its resistance to any sort of um, therapeutic, or how is this important to public health to understand this.
It's very interesting to me because I've always, I don't know, kind of had a perspective where I could think like a microbe, and people are make fun of me for that. But our agenda means nothing,
Hmm.
and it's very hard to take a perspective of a microbiological entity at all. And people always say things like a virus will evolve toward, and I get really nervous with that. There needs to be a period in that sentence. The virus evolves, and then that's it.
Exactly.
And so one of the really wild and good things about having to deal with a pandemic in the way that we have is we now have tools available, so we can start to apply those tools if, and we will, for good or for ill, other pandemics happen as well.
Hmm. Yep.
For example, we might be able to use, there are many people studying influenza and watching how it changes over time, of course. But now you have a workflow.
Right.
And there are other researchers doing this kind of thing as well. And I'm guessing as you watch the peaks and valleys and changes that take place over time, you might be able to see patterns that'll give us some insights into what's going on.
Yeah, I can show this figure from some of the metagenomics work that we're doing. So when we started doing wastewater surveillance, we were targeting specific um, viruses, mostly viruses. So we were looking, and what we still routinely do is influenza A and B, and RSV, and respiratory syncytial virus, and COVID, SARS-CoV-2. And that's changed over time. When there's certain outbreaks, we might pivot or add another target, mpox or polio or something else, depending on what we can and want to do as we partner with the public health departments. But we've also started doing some deep metagenomics, and this kind of harkens back to where I started, like pre wastewater surveillance, but looking at complex communities by sequencing them really deeply to see who was there and what they were doing, and how that was influenced by their environment. And so we're doing this is in partnership with some colleagues in Boston and Missouri and Wisconsin, and we're doing some super deep sequencing of wastewater. And you can see in this figure, it's colored by these are just viruses that are associated with human infections, and whether they're of diarrheal or respiratory or veterinary origin. There's a lot in wastewater, and I always say, like, we're when we're quantifying certain genes, we're looking for very specific organisms, but it's all always there in wastewater anyway. And so if there were ways to be able to monitor everything at once, we could learn a whole lot, and think about maybe viruses that we hadn't even been thinking of as being as showing up in a community. So it's really incredible what we can do with a lot of sequencing. Yeah.
Well, if you think about how a standard ecologist would study things, they can't really measure everything that's there with a magnifying glass and a counter. You know where I'm going with this.
Right.
But with molecular tools, you really can see everybody who's there.
As depending on how much money you have and how deeply you can sequence. I mean, that's what's tricky, is that you theoretically can see whatever's there, but some of these aren't that abundant. And even if they're like ecologically relevant bacteria or viruses or archaea that are present, if they're there in low abundance, we might not capture them in sequencing. It's sort of, the analogy is like, having a jigsaw puzzle and blindly reaching in and pulling out a couple of pieces and seeing, or mixing many jigsaw puzzles together and pulling out pieces and seeing how many different puzzles um, I'm losing my words, how many different puzzles are there in
Oh,
the
that's good.
puzzle.
Yeah.
Yeah. And so, yeah, I think it can be really hard if these viruses are really low abundance. We have to sequence pretty deeply in order to capture them. We've been doing a lot of work in my lab with ways to target sequencing or treat the samples beforehand in such a way that we sort of artificially enrich in different uh, organisms that we're interested in looking for. But you're right that we can theoretically target or see everything that's there, but practically it's a little bit harder.
Oh no, I agree with that. And we've talked on the podcast before about very rare members of a metagenomic
Hmm.
community. You have to make sure, speaking of omics, that you're not looking at ketomes, right?
Right.
And when I tell people that story, they don't believe it, but it's a microbial world. And you're looking at very rare events and
Mhm.
very rare things. So you have to do the controls carefully.
And in wastewater, you have not just the microorganisms, but you have genetic material from humans or whatever
Yeah.
else gets introduced into the system. And so sometimes that takes some bioinformatics trickery. So like if you put your samples through like the BVBRC, there's a metagenomic pipeline, and you can selectively tell it to remove certain host organisms, like don't consider the human sample, don't consider the human DNA. Let's just look at the bacterial DNA. But yeah, it's really a signal to noise issue. Like trying, which is sort of fun, right? Like it can be frustrating, but it's also fun in finding the needle in the haystack, finding something that's
It's, it's the
useful.
needle in the gene stack.
Exactly. Amazing.
But I wanted to say, you mentioned something, and I'm really hesitant to bring it up with you because the current world depresses me deeply.
Same.
And I don't want to go there, but I do want to make sure or ask your thoughts about funding of the kind of work that you're doing. This isn't looking at how stuff that I think is very important, like studying limb regeneration in amphibians. And someone can say, oh, I don't care about amphibian limbs. And I understand them. I don't agree with them, but I understand them. But this kind of thing, being able to monitor things that can cause disease through the wastewater, would seem to be a real priority. Are you getting trouble with funding issues now?
Not at the moment, but we're always on edge since January. We've been on edge. We A lot of the routine surveillance work that we do is federally funded, but comes to us via the local public health departments,
Yeah.
state and city public health departments. Um, it's just uncertain. We don't know. We Another project that I have that's federally funded. Uh, we met with the program officer recently and asked about the same question that you just asked, and she was like, we can't talk about future fund. We just don't know. I don't have a good answer, but all we can do is carry on with what what we can with the resources that we have, and know that this work is benefiting people. We have on our we have a public facing dashboard for this, and people can reach out. There's an email address that people can reach out to us, and we have people who comment that they have no other source of disease levels in their community other than our data, and they rely on it. They catch blips and they ask us questions, and so knowing that it's valuable, it's knowing that it's valuable to our clinicians and public health partners, keeps us doing this.
Rachel, on some of the other podcasts at microbe.tv, like This Week in Virology, I've heard rumblings that though you can use this approach in wastewater to monitor the progress of poliovirus, the CDC doesn't report that kind of data. Do you have thoughts on this, or do you know that to be true?
Yeah. Unfortunately, I think that is true. It's very restricted about what you can measure. And I think it's not just CDC. I don't want to just throw our government under the bus, but worldwide there's a whole network for polio surveillance, and in some countries it's routinely done, polio surveillance. Here we've done polio at the direction of the CDC. When a couple of summers ago there was an outbreak, there was a case of paralytic polio in New York, and CDC was tracking any connection between that community in New York and other cities or locations around the country, and Chicago was one of them. And so they asked us very specifically to look for polio. And so that was a case where we could, with their blessing, do it, and it's actually been published. So that paper is out. But routine surveillance is not something that they support. And I think a big reason for that is just having to deal with any sort of, like, how do you respond to the findings? Because with polio, there's lots of things you can do with this wastewater data, lots of actions you can take. When you find polio, really the thing to do is to push vaccination. That's what we can do, and we can also keep an eye out for possible clinical manifestations. But I think it just takes this mobilization of a huge response by local public health departments all the way up to the CDC should polio be detected. And so there's a little bit of hesitancy to start that. But I think, Vincent is always saying that if we did that, we'd have a baseline and we would know whether it's how much is there and how that changes. But it just takes this massive response that makes it challenging.
Every week I put together some useful links, and Rachel, you've been great about giving me lots of things to put in there. Not everybody does, and thank you for that. So folks, if this work interests you, and it should, do check out those links. I know everybody is every week anyway, of course.
Right. Yeah, I think some of it is just our public facing dashboard that you can see. I did provide a link to the BVBRC, which is something that I've gotten in the last couple of years really passionate about, teaching any sort of computational or bioinformatics skills, because you can be a really good biologist, but when we're generating this sequence data, if we want to understand it, we have to have the right tools and right skills to be able to make sense of those data. And so there's a link to that too.
Even though I'm only partially present anymore, I always wanted to learn more about bioinformatics. I actually signed up to take one of the K-based courses, so
Those
I'll
are
keep you
great
posted.
too. Yeah,
Yeah.
we're hoping to develop some similar courses for educators for the BVBRC. So keep your eyes on that. It's a work in progress, but
So let me pique your interest. Many years ago, I got a metagenomic library from Joe Handelsman's lab made in a cloud forest in Puerto Rico. And long story short, for years, I've had students interrogate that library for drug resistance genes, and they get plasmids that carry these drug resistance genes, but I never had the facilities to sequence them to figure out what's there. So you might be able to direct me to some people who could do that.
to do the sequencing.
Yeah, because here's the thing. Can you imagine a student isolating something themselves? Let's just say it turns X-gal blue, staying away from AMR for a second.
Sure.
And they don't know what organism it's from, and they don't know if the gene is related to other beta-galactosidases. And getting that sequence and analyzing it out of something that's only 150 kilobases would probably be a good learning experience.
I think it's fantastic. I know some people who use sequencing in their classroom. I haven't been able to implement it here yet, but I think it's such a valuable tool. I do bring in old sequencing gels and then some examples of some MySeq or ion torrent chips, and I show them how we used to do sequencing and how it's done now. But I think having that hands-on experience and then getting the data and then being able to analyze those data is so valuable, and it also gives people the skills that make them marketable for whatever is next. I mean, people are always looking for folks with any sort of computational experience, data analysis.
No, absolutely. And, very few, I'm at a small liberal arts institution, and we now have a bioinformatics course taught by one of my colleagues, and that's great.
Hmm.
I think it needs to be in most of our classes now on some level.
Hmm. Yeah.
And, plugging 16S sequence into BLAST is not going to do it. We need to do better than that.
Yeah. It's a start.
It is a start.
Yeah.
Well,
And
this has been really wonderful, and I love how you've talked a little bit about the way that you came to this field. And I want listeners and viewers to remember, I say this each week, that many times people have a preconceived notion about what it takes to become a researcher in the microbial sciences. The fact is, you have to be open to your curiosity and open to opportunities.
mhm. That's the
And
best part.
Pasteur said chance favors the prepare mind, and and it's true. It's true. It's probably more elegant in French, but the point I'm getting at is that there's always things out there, and you have to be ready. I mean, I've had so many people on this podcast that did not know, for example, they wanted to be a microbial scientist, and you would have never guessed while you were a postdoc that you would be doing work with wastewater.
I know, and I think my whole path is a little bit circuitous. I knew I wanted to do microbiology, but I didn't know in what context. But I think, that's the most fun thing for me, is that I follow wherever the science takes me, and I ask questions, and I'm able to explore. And if it provides information that's either useful to other scientists or useful to the community or useful to the public, that's an added bonus.
So, if you were to tell listeners and viewers just a couple of things to remember about what you've talked about today, what would those things be?
Well, so wastewater is actually, you forget it when you flush your toilet after you take a shower. You don't think about it, and most of us don't think about it all the time, but it's such an important microbial system, both in the treatment, in shaping the environment that it enters, and in telling us about something about the health of the community that it comes from. So I always say, often when I give a talk, I thank the audience for all their anonymous donations to my research. So everybody who participates, we don't target individuals, and we can learn about the environment, we can learn about community, and we can learn about, most importantly, maybe the microbes.
This is so interesting that you say that, because often if you're going to look at a particular if you're talking about gut microbiomes, for example, you have to pay very careful attention to the proper way that you approach that to preserve the anonymity of the donor.
Yep.
You don't have to worry about that because everybody's a player.
Exactly.
So you get a flush, and you get a flush, and you get a flush, to quote Oprah, right?
Exactly.
I'm
And I
sorry.
won't know it's you.
That's just that's right. Although I've seen Gattaca, the movie Gattaca, and they'll be able to figure that out soon too. But that's the strange, brave new world in which we live.
Right.
I really appreciate, number one, how kind you've always been to me. Thank you. Number two, your enthusiasm that you really brought to the forefront. I've been so pleased with what you've had to say. You've been such a wonderful guest. I also think that what you talked about today is really useful to people. A lot of times folks like to battle one another, especially nowadays, but we can all agree, I think, that understanding the march of microbes through a community as measured in wastewater is an important thing to follow and learn from.
Thanks. Yeah, thanks. And thanks for having me. Mark, you have always been such an inspiration to me, not as a, I teach, but I'm primarily a researcher. And as I, you know, try to be the best educator I can be, I look to people who are like you, who are so great at what you do and share that enthusiasm with people. And I try to take that and use it to communicate to my students and anybody that I talk to. And it was such a delight to chat with you.
Oh, I have such fun with this kind of thing. Here's a little secret. I do this podcast for me as much as anybody else
I bet.
because I have such fun with it.
I bet.
I want to wish you and yours the very best, and you'll be getting a package shortly with thank yous. And and I wish you the greatest day.
Thank you. You too.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with especially epic links this time, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Rachel Poretsky is in the Department of Biological Sciences at the University of Illinois, Chicago. Guess where? In Chicago. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.